Now, how about ER beta in this brain area? So in next few slides, I'll talk about ER beta knockdown effect. Since VMN does not express ER beta, we only tested in two different areas, which is MEA and POA. Other than that, it's exactly the same paradigm as ER alpha knockdown experiment. So as you can see here, ER beta knockdown in neither brain site on both sex and aggressive behavior did not produce any effect regardless of the time of viral vector injection. However, we have found very interesting behavioral changes by ER beta knockdown in the medial amygdala, which is sexual preference in male mice. In this test, experimental mice were tested in his home cage for the preference between receptive females and non-receptive female, which is this side, or receptive female versus intact male, which is this side. Control animal shows preference toward receptive female versus non-receptive or intact male. Whereas ER beta knockdown mice did not show any preference to receptive females when they are presented with non-receptive females, although they showed normal preference towards receptive female mice when they are presented with intact males. Therefore, ER beta knockdown in the MEA produced a disruption in sexual preference when male mice were tested for the choice between receptive and non-receptive females. Based on this finding, we tested more for the mechanism of ER beta mediated action of steroid hormone in the regulation of the sexual preference behavior in male mice. As I already discussed, on or off regulation of male sexual and aggressive behavior is mainly regulated by ER alpha, whereas how male mice react to receptive versus non-receptive females to show final expression of sexual behavior or aggressive behavior is fine-tuned by the role of ER beta. In knockdown studies, we looked at receptor expression itself, but in the following few slides, I'll talk about neuronal activity of ER beta expressing cells in MEA during this sexual preference test. Again, we focus on MEA, where not only ER alpha but also ER beta are highly expressed. In the final section, I'll talk about brain mechanisms of ER beta mediated behavior regulation. I said ER beta was found much later than ER alpha earlier in my talk. This slide shows brief history of ER beta discovery and analysis of ER beta action in last 25 years. The finding of ER beta is stimulated by the development of ER alpha knockout mice. In these knockout mice, back then we only called ER knockout, produced a lot of finding that estrogen can induce some gene expression in knockout mice. So this stimulates the finding of second estrogen receptor. Now we call estrogen receptor beta. Soon after this discovery and cloning, ER beta knockout mice are developed, as I mentioned in my behavior study. And then, for a few years, it's very good for the ER beta studied because very powerful ER beta antibody was created, and ER beta distribution, which is very different from ER alpha, was identified. However, starting around 2010, there was no good ER beta antibody were available in entire world. So we could not progress ER beta study any further, mainly because we didn't have any good ER beta antibody to identify where exactly ER beta is localized. So recently we created ER beta RFP transgenic mice in which ER beta expressing cells are markered with red fluorescence protein. As you can see here, here is a ER beta in messenger RNA expression. This is lower and higher magnification. We looked at the co-localization of this ER beta messenger RNA with red fluorescence protein in this transgenic mice. This is a slide section of the paraventricular nucleus, which has a lot of ER beta expressed. As you can see, expression of RFP and ER beta are completely matched. So we conclude we can use these mice to study the distribution of ER beta. We already knew that in medial amygdala, both ER alpha and ER beta are expressed, as I showed you in few slides back. However, we did not know where exactly ER beta is expressed in medial amygdala, and also whether ER alpha and ER beta are expressed in the same cell, not in the same region, whether it's the same cell. So this is, we found ER alpha expressing cells, ER beta expressing cells detected by this RFP protein, and we merged both of them. As you can see, medial amygdala, posterodorsal, anterior part, ER alpha is predominant, whereas posterodorsal posterior part, a little caudal part, both ER alpha and ER beta express, but especially in this area, ER beta are highly expressed. So we targeted this ER beta to see the role for male sexual preference. To manipulate neuronal activity of ER beta expressing cells, we created another transgenic mice, which is ER beta Cre mice. And with the use of DREAD system, we specifically inhibit neuronal activity of ER beta expressing cells in the MEA by injecting this virus to both sides of the MEA. And then we tested the animal for the preference between receptive and non-receptive, and receptive female versus male choice, as I showed you before. Under the test with saline injection here and here, in which ER beta expressing cells neuronal activity is normal, all male mice showed preference to receptive females, as you can see here. This is a percent of preference toward receptive female against non-receptive female, which is more than 50%. And here. Whereas in the second test, when we tested with CNO injection, in which this dread system specifically inhibit neuronal activity of ER beta neurons in the MEA, disrupted receptive female preference in male mice, which is male mice sniff both receptive and non-receptive females about the same percentage. In contrast, the same treatment for the choice between receptive and male mice did not affect at all. Male mice show the preference toward receptive females when they are tested under saline treatment, this one, or CNO treatment. As you can see here, all three conditions, they showed always preference toward receptive females. The higher means receptive female preference. Furthermore, we recorded neuronal activity of ER beta positive cells in the MEA while the male mice sniffing receptive females with the method of calcium imaging. Although I will not go into the detail of the method. As you can see, here is a receptive female corner. This is a non-receptive female corner. The male mice, when they approached receptive females and sniff here, the neuronal activity was much higher. As you can see, it's darker red dot compared to when they are vicinity of non-receptive females. As you can easily imagine, this ER beta neurons in the MEA is not the only one who can produce this kind of behavior phenomena. So we looked at where these ER beta neurons project inside the brain. And we found most of these ER beta neurons in MEA PDP send a projection to principal nucleus of bed nucleus of stria terminalis. As you can see this slide, ER beta cells in MEA project actually ER beta cells in this BNST. As you can see here, all these red are ER beta positive neurons in the BNST, and green fibers are the projection from MEA. To summarize, MEA ER beta neurons are very important to filter female information which is receptive or non-receptive females. And the information filtered in ER beta positive neurons in MEA is sent to BNST. On the other hand, how male versus female information is processed in the brain is not exactly known at this point. In terms of this female information processing, how about after BNST, where this information may be sent for male to finally show the preference behavior? When we look at the projection of ER beta positive neurons in BNST inside the brain, we found actually they send back the projection to medial amygdala posterodorsal part, where a lot of ER beta were expressed. So there is some reciprocal connection between these two areas. Also, BNST send the projection in a lot of brain area, which showed here. These are all part of social behavioral network for male sexual and aggressive behavior. Now, few more slides to conclude my talk. As I said at the beginning of my talk, steroid hormone has lifelong action throughout our lives, and the finding I explained today was based on animal study. But as I said in the beginning, finding in animal model can explain human behavior to some extent. Of course, human brains are a lot more complicated, and also environmental experiential influence is more complex. However, by looking at time, sex, and brain site-specific action of hormones throughout a neural network for social behavior, we should be able to understand social behavior in human beings. As I said in the beginning of the talk, being social is very essential for our life, but we have a lot of issues like antisocial behavior such as suicide, asocial state, escalated aggression, and also lack of pair, motherly, and fatherly bonding. So, by investigating how hormones act on the brain to regulate social behavior may help to solve this problem and understand social behavior in human beings. Finally, not only my own laboratory, but also through International Education Research Unit program at University of Tsukuba, we are collaborating with Dr. Larry Young, who is world-famous scientist in social neuroendocrinology. So if you are interested in our work, please contact us. This is the people who contribute to today's talk. And finally, if you have any question, I'm happy to answer. Thank you.